peer-reviewed scientific journal that publishes experimental methods in video format. And it is the world's leading video platform for research and education. It creates a video that showcases how peer-reviewed research methods can be performed in the lab. So it's a completely unique audience of instructors and researchers. So you'll discover how visualization improves research reproducibility, reduces trial and error, and streamlines lab techniques. So I really need to turn it over to Alice, who is the Director of Brand and Public Relations at JoVE, because she is the expert on this. And this is one session that I will be learning just like everybody else, because I have very little knowledge of it. So please, Alice, you can go ahead now. Thank you very much for joining us.
Thank you, Lenny, and thanks everybody for coming today. I'm really excited to be presenting. I am going to share my screen just to make sure that we can all see and hear what I am doing. So please let me know if this is visible. Is it all fantastic? So again, warm welcome to everyone. My name is Alice. As Lenny mentioned, I am the Director of Brand and Public Relations at JoVE. Today's session will be a little bit of an introduction, because usually what I find myself doing is actually diving deeper into pedagogy techniques and doing some more in-depth analysis of, how we can leverage video tools for teaching and research. But because many of you might not be familiar with JoVE, I thought we would start from the very basics just to get all on the same page. And then we will have time for questions, for deep dives, or anything that you might find relevant. One disclaimer, JoVE has been a partner of Research for Life for quite some time. We initially provided access in 2013, but only this year we managed to join the partners meeting and be part of the Research for Life friends who have supported the cause with a donation as well. And one of the things that really struck me when I met some of you in person was that I realized that pretty much no one knew what JoVE was. And for a brand director, this is, quite telling. But also, I felt like many of you probably didn't realize how useful it could have been for their own scope and needs. And so that's what we're here to do. First, to establish a little bit of recognition of what we do. So next time you hear about JoVE, you will not be surprised to know what we do. And second of all, because I would love for you to be able to leverage these resources. As I mentioned, we've been partners of the Research for Life Foundation for over 10 years, and it would be a shame to continue to have this gap between what you can access and what you're actually able to use on a daily basis. So, without further ado, what is JoVE? Well, as the introduction mentioned briefly, we produce videos. We're a producer of videos, but not any video producer. At the moment, we are the world-leading provider of science videos, yes, but the mission is much broader than just creating videos. When I say videos, people think about YouTube videos, think about maybe documentaries, they think about a lot of different things. So it's good to specify what kind of videos we create. First of all, we have videos on both research and education. And that means that there is a journal publication. So JoVE started off as a journal. And from that publication, we, of course, publish the papers through a normal peer review process. And after the peer review, after the papers have been accepted, we then create a video out of them. How does that happen? There is an actual video production process that takes place. So the lab that created the procedure and decided to publish the paper will be provided with a script that we create internally, and through a series of feedback loops and rounds of research done on the papers themselves, we extract the bit of the paper that we believe could be produced in a video format. This is how it started, right? That video is then recorded and subsequently published by actually going to the lab, to actually replicate the procedure that's been explained by those who created it. This is, of course, very important. We'll see a variety of different applications of this technique of recording people who actually made the papers and who actually created the procedures in the first place. But that's one kind of videos we produce. The other kind is the so-called video textbook. So these videos related to education, they are much more focused on concepts rather than experiments. These will be both animations and videos that have been recorded at labs, through clinical technicians, through any sort of real life setting, but they are aimed at educating. So instead of just being related to research, those can be suited for all levels of higher education. Sometimes even we try to propose them to students who are at a turning point in their education. They're going from secondary education to higher education. That is a great spot to be if you want to learn core concepts. All of these videos together, they make up our library of over 25,000 videos. So later, when we go onto jove.com and we browse through the platform, you'll be able to navigate and search for videos. There will be over 25,000. And every year we continue to publish thousands of videos, both in the research area that you can see in the preview and in the education section. There is a third section that's very new called the business section, which is why when we say science videos, this might change one day. We're kind of pivoting towards saying STEM plus, so science, science, technology, engineering, and mathematics plus, because we are including more and more videos related to business education, humanities, psychology, and many more. So all of these together, 25,000 videos, how do you access them? Through the Research for Life platform. Again, we're going to see how to do that, but together with you, there are almost 1,400 subscribing institutions worldwide that access JoVE videos. Of course, you have a subscription through the Research for Life portal. In many other cases, institutions decide to come to the JoVE platform, they buy a subscription, and they're capable of showing this to all of their students, all of their researchers, all of their faculty members. Now, why is that important? Because it creates for a positive feedback loop. If there is something that needs changing in a video, if there is something that lacks accuracy for whatever reason, maybe the automated subtitles were not great, maybe we need to put some changes in place, maybe we need to make some progress, having top-tier subscribing institutions worldwide is very important. It allows us to gather feedback, to create new products, and a lot of the new things that we create come straight from requests of these subscribing institutions. So it's not the individual who accesses the platform by paying for it, it's always the institution. So it's always librarians, it's always decision makers around the world who say, okay, I would like to have this resource for my community. And the same happened here. You have access through Research for Life because we've been partners with them for a really long time. So you can make the most of all of these videos. Just to give you some more numbers, these huge ones that you see here, 66 million unique visits. It's just to give you an idea of how much these videos get watched. And if you think about it, in 1 year, cumulatively, all of the visits, basically almost 343 million days were spent on the website in 2023 only. So it's just to give you an idea, a ballpark figure of how much people commit to watching these videos. Why is that important? Again, because videos are a wonderful way to learn. A lot of us, if asked, would say, I am a visual learner. That is true for many people, but it's especially true when we do research because visualization really helps us understand concepts much more quickly. Without, of course, compromising. Because when we have videos, it doesn't necessarily mean we shouldn't have any text or we shouldn't have anything to the side. It means we are complementing the access. So when something is hard to read, the readability is low, the text is still there, but the fact that there is a video that goes with it makes a world of a difference. And it's what I always say, it's like watching a recipe a video recipe rather than just reading the recipe, especially if somebody's fond of baking. It's full of rules. It's pretty much chemistry. If you don't know how to apply some of the techniques, watching them being performed makes the world of a difference.
This daughter strand.
And that's exactly what we can see here. So
This daughter strand is known as the lagging.
Pauses. I'd like for you to take a moment to read with me what's written on the left. So this is a very, very typical line that people would find if they were studying molecular biology at all levels. And this is about how the daughter strand, I'm going to read it with you, known as the lagging strand, can be replicated. So DNA polymerase can only synthesize DNA in the 5' to 3' direction. Because of this, the leading strand is synthesized continuously. However, DNA polymerase cannot synthesize DNA in a 3' to 5' direction on the lagging strand. To deal with this problem, DNA synthesis is carried out discontinuously in a 5' to 3' direction. Now, some of this is jargon, DNA polymerase, 5' to 3', but think about how difficult it can be to understand what the direction is and just how the synthesis happens through this text. Yes, of course, some textbooks, a lot of textbooks have images, but now let's watch the corresponding video with the audio on top to see how different visualization is.
This daughter strand is known as the lagging strand. DNA polymerase can only synthesize DNA in the 5' to 3' direction. Because of this, the leading strand is synthesized continuously. However, DNA polymerase cannot synthesize DNA in a 3' to 5' direction on the lagging strand. To deal with this problem, DNA synthesis is carried out discontinuously in a 5' to 3' direction.
This was just an example, but you can already see how different it is to see something with your eyes compared to just reading it. This again is a very simple example from the education sector where you can see how a simple video can help us understand a concept in molecular biology. The same applies to research. And that's why we started making videos in the first place. The idea was, why don't we make it easier for people who have created a methodology to disseminate that methodology, to explain that methodology, and one day to have it replicated. So in turn, for the scientific community, that means advancement. That means fewer resources wasted. That means reducing costs. In a constrained environment, this is so important. It's important for everybody, but in a constrained environment, it's even more important because it means we can take whatever we have as a methodology and replicate it almost instantly. So let's see some of the issues that JoVE is able to solve through this process. When it comes to research, which is the one I was mentioning just now, we often have a high cost associated with certain research methods because the reagents cost a lot of money, because maybe time itself is a cost. And learning new methods, so reproducing them, is often a constraint. It's often an issue. What happens all the time is that new lab units, or maybe people who have not been trained yet, they think, okay, we could innovate and do a different procedure that in the future will save us time. But learning that procedure, knowing how to do it properly, and the attempts that it's going to take us to get to that result, they're a waste of time right now. We don't have the means to innovate, to do a different procedure. And so what happens is, because of the reproducibility issue, oftentimes we get stuck with outdated procedures, outdated methods, and we're not able to innovate. Or more simply, when we want to try to do an experiment and we read the paper, we're not able to see how that plays out in our lab, then this can be an issue. We try to get in touch with the people who created the paper. We ask them to film something. It's never going to be the same. So in this case, having a video that shows you exactly how to do something addresses the issue of reproducibility. You see, and you can replicate. Again, the devil is in the details, especially in research. The fact that something needs to be stirred in a certain way, the fact that the pipette needs to collect a micro pellet at the end of a test tube. These things make the world of a difference. And so knowing how to do it because you've seen it done in video, I guess many of you have seen it throughout the internet with unreviewed papers, it can make a huge difference. In this case, all of the papers are peer reviewed, so you have that added security of knowing that what you're reproducing is valid. Then the issue of productivity. Of course, when we have new lab members, when we need to train new people, one of the issues is related to the fact that for a certain period of time, their productivity is going to be quite low. They're not going to be as productive as other team members because they need to learn. The fact that you can transfer knowledge very quickly, then it means that productivity can go up while reducing the costs. And of course, limited funding to train, learn new methods connected to that, very, very strictly connected to that. And last but not least, the loss of knowledge. When somebody leaves, when somebody needs to go to a different lab, a different country, a different reality, oftentimes we think, okay, we're going to lose all of the knowledge associated with that person's skills. Filming that person is one of the main ways to retain that knowledge. And so similarly, if you have papers in video, if you have research being captured in video professionally with a high quality, that means the staff can maintain that knowledge within the lab without having the issue of turnover. As for the education side, I know many of you during the Oxford Partners meeting were interested in the education aspect of this. Of course, there's a variety of things that videos achieve, especially when we think of younger students. One of the issues is related to engagement, the fact that they might not be engaged with the material, with the course material, with the content that we create. Some of it might be lengthy, some of it might be perceived as boring or as difficult. The fact that you alternate using a video, it increases the engagement. That's just natural. It's how people use their phones nowadays. It's how they use the internet nowadays. Most students will find videos beneficial for their learning journey. And when it comes to complex concepts, as we saw earlier with the lagging strand in the DNA synthesis, the issue is that oftentimes science cannot be seen. Oftentimes it's too tiny. Oftentimes it's too abstract to be visualized internally. And one of the things I talk about the most in my webinars is the fact that everybody is a different kind of learner. We give for granted that people learn the same way as we do. And often lecturers, faculty members at university, they've not received pedagogy training. And so they don't know how to transfer their knowledge to somebody who doesn't have necessarily the same way of learning, the same skills, the same set of skills. And so if the complex concepts are hard for them to grasp, again, videos can be massively helpful. And when I say videos, I mean not just for the visual purpose, but also for the auditory purpose. The fact that all of the videos have subtitles in different languages, so they can be immediately translated. The fact that the audio is very self-explanatory. And there is a great case study from the University of Milan of a blind student who actually managed to graduate learning through the videos, which can be counterintuitive, a blind person, visually impaired person, watching videos, playing videos to learn. Well, it is possible. If the audio is very, very highly descriptive, that's absolutely possible. It can help the understanding of those concepts. Another issue that a lot of institutions face is low student retention. Science is difficult, STEM subjects are generally very hard, and so having a higher retention rate of students can be particularly important when it comes to understanding exactly how to make sure that the students feel supported throughout their journey. And so that turnover that can happen with students as well, if we have more than one way to make sure they come close to us and to give them material to learn, that also increases their attention long term. Poor test scores, that's one of the main issues. Students retain the concepts for a short period of time, and then when they get tested, it's a nightmare. They don't know how to do it. Those test scores can definitely be improved if they have a variety of means to learn, one of them being videos, of course. But on the JoVE platform, you will also find quizzes that can be used for formative purposes and that can be used to test knowledge before going to the actual test, to the actual exam rooms. And finally, a lot of people in higher institutions, higher education institutions, have little time to prepare course materials. Many of them are dedicated to both research and education. Many lecturers don't have a lot of time to refresh the content every now and then. And the fact that easily accessible content is of high quality, it's been peer reviewed, it's been created by scientists and by fellow scientists, it makes for a great starting point if you don't have a lot of time to prepare course material. And to show that, I've put together a couple of case studies that we've created over time. This one in particular was made by showing JoVE videos to a group of students at UCLA, the University of California, Los Angeles. And what we saw in the group, in terms of retention of information and test scores, was that students tried to take tests before and after watching videos related to a specific subject. This was an independent study conducted at UCLA. What we saw is that the test scores improved dramatically because not even 30% of the students were able to pass the test just after verbal explanations. But after watching the videos that came with a verbal explanation, their retention improved massively, and it went up to almost 90% of the students understanding the content and getting test scores that were averaged to pass the test. And even more significantly, a case study that comes from the University of Pavia. So this is a project I personally worked on in my early days at JoVE during the COVID period, during the pandemic. So at the time, of course, visual learning was already something that many universities, many institutions were trying to leverage, but the need was definitely lower because there was just no necessity to find other ways to learn that instead became prominent during COVID. So imagine we have this institution that comes to us and says, we've had a JoVE subscription for a while, it's wonderful, but now we have an added problem. Our students in clinical skills cannot access the hospital. They cannot come to the hospital. How can we train them? How can we do a traineeship that is fully virtual so that when and if they're able to come back, these medical students, they're not completely lost. They haven't lost a whole year of clinical skills practice. And so we created this project for them. We called it a special project, but nowadays we do so many of those that it can just be applied to any scenario. Basically, we created a list of playlists and videos made specifically to help students understand how to work with patients ahead of working with patients. So in that scenario, it was compulsory. They just couldn't go into the hospital and work with patients. And so what they did was they watched these videos and then as a full-on tutorial on how to be a doctor. Again, how is that different than going to other platforms, YouTube? These videos have been created by the people who practice these techniques. And so through that, what we were able to do was we asked them, is this going to be helpful when you then go back? So watching the video is great, your level of confidence seems higher, but can you please give us some feedback once you actually go there? And the results were astonishing. So good that 5 years down the line, all of the students are still watching these videos as a compulsory step in their training. So even though the pandemic is over and the issues connected with the access to patients and hospitals have been resolved at the university, what continued to happen was that they took that practice of watching the virtual traineeship program to then go on and apply it, and they continue to do that. They notice that it increases their confidence, their practical skills, their ability to actually talk to patients, work with patients enormously. So now this is not a special project anymore. It's just something that we do. It's a great example of how you as well can take the videos and can apply them straight away. I can imagine, again, for many of you, many of the country connectors, this will be an issue, right? Accessibility of patients, people that we can do traineeship on. Watching the videos again doesn't replace that, but it puts people in a simulation setting where they can then go and apply. And it doesn't have to be asynchronous. It can be synchronous. You can still watch the video and apply it. Remember when I said the recipe and the video recipe? That's exactly what we would do if we were cooking something. We would watch it and repeat. And similarly, students, medical practitioners, they can do that. A lot of the techniques that you find in the journal section, this was a project for students, but it can be applied to anybody. A lot of the videos that you see in the research section under medicine or clinical sciences, they are great because they show high impact procedures, cutting edge methods. These can be directly applied by practitioners. And so just an idea of how many applications there are in this sector. All of these can be applied if you then go and, listen, go and see what can be useful. I think the main point here is understanding what your community needs and trying to propose different things according to what your community might require. So let's have a demo. What I'd like to do with you right now is browse through the platform. I know this session is recorded, so you can do what I just suggested. You can just watch it now and then later apply it asynchronously. You can do it with me, but my recommendation is to follow it right now with me. And then later, when you've got a little bit of time, browse in your own time. If you need to rewatch, I believe the recording will be available to anybody, so you can take a look and you can see all of the new answers. So let's go on to JoVE. So very simply, if you wanted to browse through the JoVE platform, jove.com will lead you straight to where you need to be. This will be
Alice,
Yes.
can I interrupt you? We have a question.
Absolutely.
Yes, I forgot to say that would be my job too. Greetings. I need some clarification. These
Yes.
videos on JoVE, you cannot access them individually, but rather an institution must pay a subscription for all the students in order to access them. Is that
That is
correct?
correct. That is correct. However, Research for Life has access to this. So if you are affiliated with Research for Life, you will be able to watch all of the videos through authentication through your website. Yes.
Yeah, I wanted you to clarify that, please.
Yeah,
Thank you.
absolutely. Thank you. Thanks to whoever asked the question as well. And for those who are not part of the Research for Life group for any particular reason, yes, that's the way one subscribes. It's not we call it a B2C, so business to customer model. We don't activate it with individuals. It's a B2B, so business to organization model. So any institution can access JoVE through that process. So this is a homepage you will all find if you just go onto jove.com. The most important thing for everybody is to go onto the sign in area, okay? Otherwise, everything I said will be inapplicable. So once we go to the sign in area, there we go. You will be required to add your institutional email and password. My recommendation, because you're affiliated with Research for Life, is to continue with either a single sign-on or with Google, depending on how you authenticate to the main platform. Now, in my case, I have a JoVE account, so I'm just going to add it here. But again, if you're experiencing issues, you can also just create a new account using your institutional email address. If you experience any issues, you can always get in touch with us. There is a contact us button down below where you can simply contact our support team, and they're going to be able to redirect you to the correct instructions. So sign in. This is what I'm going to do right now. Okay, wonderful. So we are onto the main homepage, and we can already see that there's a few things: most watched research videos, most watched education videos, just to give you a glimpse of, the variety of content. But my favorite way to browse through this platform is either going to the search bar here or browsing through the 3 drop-down menus: research, education, and business. I'm going to give you an overview of each of these sections, but first of all, why don't we just go for a search? Does anybody want to propose some keywords, something they'd like to put forward? Let's see if there's something in the comment or the chat. Don't see any suggestions yet, so I'm just going to go with something that can be, for instance, oh, let's see, somebody suggested something. Bat virus, wonderful. So why don't we search for bat virus and see what it gives us? Okay, anytime we do a browse, we're going to get a mix of research and education videos. If you want to filter for one or the other, because again, their purpose is very different, we can just go for research only or education only. Alternatively, what you see here is an icon, in this case, research, that tells you which one is the area that you're looking at. Being bat virus a concept that's primarily used in research, I'm not surprised to see that the majority of the papers are related to research. And again, many of the things that you find on the left can help you filter these results. It can be all types of articles or those with video only. This is because I explained the main value of having a video, but JoVE is a journal, so a lot of the content will have videos. Some of the content will be other formats, such as reviews or case studies. Some of them might not necessarily have a video associated with them. So being a different type of publication, some will not come with an accompanying video. But in any case, let's keep all of them here. We can also filter by date. We can choose here if we want just the most recent papers, say published in the past 5 years. And we could even decide to search by author or institution if we were looking for something specific. So through these filters, we can then narrow down. And let's say I want to watch this fast video, mosquito-associated virus isolation from field-collected mosquitoes. So first thing I will do is open it. Our publication, by the way, is multidisciplinary, so you will find videos in a lot of different areas, which, by the way, are all collected up here as well. Behavior, biochemistry, and so on and so forth. So the video is here to the left. The chapters are simply a way of subdividing the video into bite-sized content, so it allows us to go to specific sections. Maybe we just want to see the virus isolation in this case. And then there will be some related videos and, of course, the full text and the cite button. You can also save it to your favorites, or you can change the language, which is, again, automatically generated. So there is a variety of different languages, very, very handy, very useful. It is, however, AI that generates these. The transcript and the summary are only related to the video itself. So if we want to go for the full text, again, the button is right here, and I can show you what it means to click on it. There you go. It's a classic paper. It's been peer reviewed. It's got all of the different areas, protocol, results, discussion. There's easy navigation here to the left. You can also download it as a PDF or just the materials list. And again, for citations, there's always a possibility to cite through the button, and it both gives you a citation type that you can copy, or you can export it for any sort of reference manager that you might be using. Now, going back to our video, I'm going to play it now. I just want to show you what the video looks like. Okay, so this is a random video that we picked through your selection of keywords. Let's see what it's made of. Maybe we can watch the first 2 or 3 minutes so that you can see both the introduction section and the rest. I'm going to add English subtitles so that everybody can follow. But again, there's subtitles, languages available in many, many different regional languages.
This protocol is a useful approach for isolating mosquito-associated viruses and can help in further research into the distribution of pathogens related to mosquitoes and the control of mosquito-borne diseases.
Using thin lines to isolate viruses in a biosafety level 2 laboratory is safer and more efficient. Unlike other virus isolation procedures, such as cycling rate, inoculation with very fluid. This technique does not necessitate animal experimentation or ethical assessment.
The contamination from samples and cells drying during the incubation can lead to the failure of the experiment. Hence, it is necessary to add 2% penicillin, streptomycin, and fluoxetine into the medium to prevent contamination.
So this was the introduction. Now on to the technique.
Begin with the addition of 3 mm ceramic beads, 1.5 milliliters of RPMI medium supplemented with 2% penicillin streptomycin amphotericin B solution in a 2 milliliter sterile tube placed on ice, and then transfer 50 mosquitoes in it. Next, homogenize the mosquitoes with a low-temperature tissue homogenizer by grinding for 30 seconds at 70 Hz and 4 degrees Celsius for 3 cycles. Centrifuge the homogenate at 15,000 G for 30 minutes at 4 degrees Celsius and transfer the supernatants to the new tubes. To remove the mosquito debris completely, centrifuge the supernatant again at 15,000 G for 10 minutes at 4 degrees Celsius. Aliquot the 200 microliters of supernatant P0 into a 2 milliliter screw cap storage tube and store it at -80 degrees Celsius. For the virus amplification, use Aedes albopictus RNAi deficient mosquito cell line C636 and a vertebrate cell line baby hamster kidney or BHK21 from a 75 centimeter square flask and seed them in 24 well plates separately. Place the seeded 24 well plate overnight at 28 or 37 degrees Celsius.
Now you see the level of detail and depth of each of these. They are professionally recorded, so that's why it's much better to have a video like this than just to ask somebody to replicate the procedure. And even seeing it in front of you, you'd have to have a camera with good quality for like capturing the details. For those who might be wondering, if you go towards the end of the video, you will then see the discussion, the conclusion. So there will be an area of the video where they actually get to talk about these things. I'm just not going to show them now so that we've got time to basically do other things. But the idea is all of the papers come with this kind of format. And again, only the reviews and some other types of papers don't have a video, of course. The vast majority of them absolutely do. So anything that you will find, you'll be able to watch. And that was one way of using the search bar, right? You search and then there's a few filters, some criteria. You can go and find the relevant videos. Another great way to do searches, of course, the search bar is always here on top if you want to resume your search. But another great way to do that is to go through the different sections. So you can see here, it's a multidisciplinary journal. If we want to go to cancer research, chemistry, developmental biology, each of these will have its own associated videos. One important distinction of the videos, however, between journal and encyclopedia of experiments. I went into the cancer research section. I think it might be one of the most relevant ones for those doing research in general. The journal articles are exactly what I've just shown you. If you want to go to the Encyclopedia of Experiments, this is actually a selection of specific protocols that have been selected as the most important in the field. So here you will find cutting-edge publications that have been published by laboratories around the world. In the Encyclopedia of Experiments, you find what we have selected to be the top procedures that everybody should know about. In this case, they are divided by type of cancer, but again, every single one will have its own specific division. If we, for example, want to go on to skin cancer, there we go, we will see all of the most important techniques when it comes to making progress in this area. And again, especially if you are working with new lab members or if you want to make sure everybody is on the same page, these are great videos. They're not too long and they alternate some parts of recordings with some parts of animation to help you understand all of the basic research techniques. Some animal model studies are also available in case somebody is using animal models. these are also very, very important for the 3R principle, because again, watching videos means you are experimenting a lot less on live samples, and in vivo research can heavily rely on prep time done through these videos before actually using the samples. Now, let's open one of these. And similarly, you will see the structure is the same. It's just we don't have a full text. We just have the protocol. And there we go. The protocol where everything is explained thoroughly. We see all of the different things that we need to do. Again, we can download the materials list because this is not a paper per se. We don't have the full text, but it comes from a paper. And again, you will see the source being cited just here. If you want to access the source, again, it's clickable and it redirects you to the main video. Apologies, this one. So this is a video extract basically of what we have selected as one of the top procedures to understand how to do something. And that was the Encyclopedia of Experiments. Now, back to, I would say, our homepage, which we can always find here.
Can I interrupt for a second? We have had several questions
Absolutely.
about, is there a brochure available or a flyer? Is there something that they can print to distribute at their institutions to get
Most
people?
definitely.
Okay.
Absolutely, yes. So I shared with the committee a page called a Media Valet. It's the web pages that we create for you guys. So inside of that page, which I can share again with you all, is everything. We have flyers, brochures, pamphlets. We've created a variety of different things that you can either print out if you want, or you can just disseminate via email. So yes, let me actually open it for you.
That would be great because there's a real interest, but they need to know how to go forward in their institutions.
No, absolutely.
Yeah.
So this is what the page should look like if it loaded. I'm not sure why it's let's try again. Might be something in our cookies preventing it. Yeah, this has happened a couple of times. Apologies. I will send the link again, but basically the page includes a variety of different things that you can leverage to send out to your students. So yes, it's pamphlets, it's all sort of material that can be useful. Also for, and we will see later, integration with e-learning platforms with a variety of different things. Not sure if there are more questions.
No, there's just one about login that I'm answering them. How can we reach JoVE by a Research for Life user? And I'm saying you log in and you go to the journal list. You look up JoVE as a journal. Yeah, okay. I'm taking care of that.
Wonderful. Yes, that's correct. So that's how you would go about it. And again, it's a pretty intuitive process. You just need to remember to sign in using your institutional credentials. Otherwise, if it's a personal email address, I get this question all the time. People have just used their Gmail, Outlook, or whatever accounts. That one is not going to work.
Perfect.
Awesome. By the way, there is a lot of material here as well, in case you want to have a little bit of a browse of the different techniques, the different things that you can use. If you scroll down, there's a few things that you can subscribe to, including our newsletter. You can access our blog. There is so much content that you can use. For libraries, we have a series of resources, which again, open up here. And you've got so much: the librarian guide, outreach media material, faculty resources. If we just click on this one, for instance, you will see a variety of different things: textbooks, exam prep, resources, playlists. So pretty much anything you might need will be found just browsing through the website. But again, for ease of use, I've put all of the flyers in one place, which again, I'm going to share soon. Now, on to our education section. So the difference between research and education is that research, as I mentioned, is either peer-reviewed content that has been published in a video format, or peer-reviewed content that we thought would be essential techniques which we summarized in a video. Now, education is created differently and for a different purpose. For this reason, yes, of course, you can use the search bar. You can do DNA synthesis polymerase, the video that we watched earlier, and you can do education only, for example. And this will give you a variety of different videos, there we go, that we can find related to the topic, that can explain the topic. However, my preferred way as, former teacher and when I talk to faculty members is to actually go onto the education section up here. Why is that? Because education is very rich. It contains thousands and thousands of videos. Unless you have a specific topic that you want to explain visually, I highly recommend just going to the section and browsing a little bit. So say, for example, let's change subject and go on to physics. I go onto the physics section, and again, I get presented with two different formats of videos. Core videos are essential techniques explained through animations. Science education is research for undergraduates, basically. So it is experiments being performed in labs, but very essential, very, very pivotal for understanding the subject. And they come, yes, with an accompanying video, but also context setting, explanation. They're much richer. So the core videos, if you want to explain the core concepts, 1 or 2 minutes, they're wonderful. You're maybe going through dense lecture material, you want to alternate to have that snap moment of understanding, core concepts. You want students to go more in depth, understand, contextualize science education. Let's watch a video of both types so you can see the difference. So core. First of all, core is very rich, very, very rich. In physics, I opened a section. It already contains 33 chapters. Each chapter, this is just chapter 1, has a series of videos. So in theory, if somebody was a very, very strong-willed student and wanted to learn the entirety of a basic physics course, they could do so by themselves. It's basically a textbook. So they could go from the scope of physics all the way down to chapter 33, electromagnetic waves. Each video basically gives you a little quick lesson. So why don't we open maybe magnetic forces and fields and a video about magnetic field lines. Now, this is a different sort of video, as I mentioned. It's animated. So it's been created by scientists, by professors, by researchers, but it's not a peer-reviewed video in itself. However, again, the accuracy and the content have been checked multiple times by people in the field, and there is a continuous revision process going on. Because it's part of a chapter, you will see the other connected videos here to the right-hand side, and you will see that the video itself is available in a variety of different languages, both for audio and for subtitles. I know this is very important because we're addressing regions of the world where English might not be the first language. So English, French, Portuguese, Spanish, but also areas like Turkey, Asian countries, Japan, Korea, in Arabic as well. The videos are available with the audio. There's a wonderful variety here of audio generated content, and also the subtitles available in so very, very many languages. I find this one of the most unique parts of this process. So let's say I wanted to watch this with subtitles in Portuguese. I can open the video and I'll show you the 1 minute.
Iron filings are placed near the permanent magnet. They line up with the magnetic field to reveal the magnetic field lines. The direction of magnetic field lines is defined as the direction in which the north end of a compass needle points. The pictorial representation of magnetic field lines helps visualize the strength and direction of the magnetic field. The field lines are always directed from the north to south pole along the tangent to the magnetic field at each point. Magnetic field lines always form closed loops. They are densely packed at the poles and are widely separated away from the poles. The magnetic field strength is proportional to the density of the field lines, implying the field is stronger near the poles and weaker away. If field lines intersect at a point, it implies two directions of the field, which is impossible. Hence, field lines cannot intersect. Magnetic field lines patterns produced by like and unlike magnetic poles, a straight current-carrying wire, and a horseshoe magnet are some common sources of magnetic fields.
Here we go. 1 minute, 20 seconds, very short, very to the point, and it explains one concept. It's very, very good for micro-learning practices, right? If we want to then add something to this, there's a variety of buttons below. We can either embed the video. This means putting it onto learning management systems, e-learning platforms. The process, again, is explaining some of the brochures that I'm sharing with you, but basically it redirects you to a page where there are a few links that can be copied, including a URL. This is basically to allow you tracking who can access this video, because say, for instance, a professor, faculty member wanted their students to do all of this, but they didn't want the students to authenticate, go through the page, and so on and so forth. This is a quick way to show the video to students without having them authenticate, because it will authenticate through the person who generated the code. Then adding to playlist, one can create the playlists that they want. Basically, here are some of the ones I've created, but playlists are a wonderful way to collect all of your material in one place. Again, I only have limited time today, but there is a variety of functionalities on the platform where you can personalize, customize. Platforms are one of them. And finally, you can create a quiz. So with the video comes a set of 3 to 5 questions that have been preset for understanding and checking whether the students have retained the concepts. By creating a quiz, I get redirected to the quiz platform within JoVE with a few things to specify. But once I have, I can basically see all of the questions that have been associated with this video. Question 1 with a few options. Question 2, again with its options. 3, 4, 5, and actually here there are many, many more, up to 9 questions. Each question can be completely customized. We can type in here the answers, we can add images, we can make changes, we can add questions. Again, this entire part is another world of knowledge and possibilities. It's just to show you that, again, the platforms offers a variety of different options. All one needs is a little bit of time to actually get familiar with them. But once you are, it's basically like many other platforms that you find out there with a lot of customization options. Now, I showed you one of the core videos within physics, but I also wanted to show you the other type of videos that we can find onto the education section, which is science education. Now, let's open one of these again, physics 1 and physics 2. Maybe we go on to physics 2. And we can already notice that for the same topic, magnetic fields, first of all, the length of the videos is different. We have 10 minutes here compared to the few seconds that we had, sorry, the 1 minute and 20 seconds that we had earlier. So if we want to open this, again, I'm going to show you just a tiny portion of this video. Easy to navigate here on the right. We can already see that there is an introduction. There will be a part where we see the people performing this experiment, and then there will be applications, analysis, results. This is great to contextualize what is being taught, because students, again, are not just going to learn about the principles and theory, they're going to see the practical applications of that. So if we want to go on to maybe the visualization of the field lines, first of all.
Gather the necessary materials and instruments, namely a DC current source, a plastic board mounted with several compass needles and a straight conducting wire passing through its center, and a permanent bar magnet. Observe the plastic board with a hole in its center. It is mounted with several compass needles around the center hole using pins, such that the needles are free to rotate. Also note that the conducting wire is fed through the center hole of the board. Make sure that the wire is perpendicular to the board.
Again, this just to show you that the actual practice is explained with a level of detail that allows visualizing exactly what the researcher was doing in this case. Now, this is different than a
Carol,
research video. Yep.
you have a request for veterinary medicine. If you can locate some videos on that.
Yes, absolutely. Let me see. So vet videos.
Yeah.
So why
I
don't
hate
we
to
go...
interrupt you, but when people request things, I try to get it to them.
Yeah, no problem. So if we go on to the education section, I don't think we will find much related to this because this is more related to animals. So animal research will be onto this section. We can go, for instance, onto the Encyclopedia of Experiments. And I think the best luck that we can have is onto biology. And we have all of the different types of models. Large animals, small ones, all of the different animal models. Rodent models is one of the areas where we would find the most interesting videos for sure. But again, my recommendation is always to go for keyword search. So I don't know if you have any requests for any vet science videos that we can maybe look for. Can already see that people are saying health education and nursing students, those can be found. So while we wait for maybe a suggestion on veterinary medicine, why don't I show you both the education and the research videos for both, for nursing and health sciences. So education, as I was mentioning earlier in the case study as well, we have clinical. So if you open the clinical section, you will see we have nursing, medical surgical nursing, anatomy and physiology, pharmacology, and clinical skills. These videos, being core, they will explain the core concepts. So if we want to go on to nursing, for example, different chapters, different things, but they will all be those videos, 1 to 2 minutes, that explain the core concepts. For instance, if we want to go on to the nursing process, we will see things like data collection, diagnosis, and so on. If we wanted to see the practice of nursing and health sciences, then again, onto the clinical section, but down to clinical skills. So that's the difference between the two videos. Core videos are animated concepts, the theory, clinical skills, the practice. So physical examinations 1, 2, and 3. Then we would have emergency care, nursing skills, COVID procedures, and physical examinations again. If I go on to nursing, for instance, which is what has been requested, preparing and administering topical medications, I can open this. And similar to what we saw earlier, I'm just going to remove the audio so I can speak on top of it. We will have an introduction, preparation, and then applications. So administering the topical medication, for instance, we jump onto the section. We actually see the nurse performing the procedure with the patient, communicating with the patient. So basically doing all of the different steps as if we were in a tutorial to show what it means to perform this. So in this case, it's a patch, topical medication. So they're going to apply the patch. And again, they're going to show all of the different steps to applying the patch. Disinfectant, removing the medication that was remaining. And again, specifies what to do with the gloves, how often changing them. All of the different parts of the procedure are listed there. And after this has been done, again, the level of detail, just wanted to show you that it's very, very thorough. And then we go on to applications. What do we need to collect in terms of data? What do we need to register? What is important? This is available for both, as we saw, nursing skills and all of the other clinical skills, including medical procedures, including coronavirus procedures, including emergency care, for example. Loads of videos for each type. Then we have I can see somebody's asking for chemistry education. Yes, absolutely. Similarly, education, chemistry, and you've got a wide choice: core, organic, analytical, lab manual, science education. Lab manual is a tutorial on how to perform basic experiments in chemistry. So again, you will find 3 types of videos. For example, enthalpy of reaction. One is the concepts, one is the instructor prep. So the person who needs to set this up in the lab will also have their own dedicated video. And then the student protocol, which the students can follow. Similarly, this is just a lab manual. You have a variety of different sections in all of the different areas. Again, it just takes a little bit of patience to browse, but it's very, very intuitive to browse through the different areas and decide which one is what you would like to go for. Okay, I can
It
see.
looks like we're caught up on the questions in chat. Did you want to put in the address of where all the flyers are that didn't display the
Yes.
URL? Or
I need to
that
look
would
for
be
it for
helpful.
a second. So if you've got other things to present in the meantime, Lenny, let me just find it so I can give it to you all.
Okay. Well, we just appreciate the in-depth overview, if we can have such a term. And I know that a lot of people learned an overwhelming amount of information, and they're going to have to go back and practice. We will distribute the recording to everyone. We had, this often happens, we had many more people sign up, but for some reason they were not able to come on today. So we have a list of 320 people to send the recording to, and Michael will take care of that. So we appreciate your time. We're getting some thank yous up there. It sure opened my mind, although I'm not a researcher or a educator anymore. And we appreciate that you went into all this detail too. Okay, now we're just waiting for the URL of where
Yes, I am
the
so
brochures
searching.
are.
If I can't find it timely, because basically what happened with this portal is that some of the pages that we published are not available to external people. So what I might do is I might just share the portal, I might just share the content of the portal separately. This could also be an option, but let me just do a quick browse because
Okay, if not, we'll just send us the information. We'll make sure all the participants get it.
wonderful,
Okay.
wonderful.
Yeah, whichever way works. And when you're not as, in the middle of this and you can think it out. Yes.
Fantastic. Meanwhile, I just want, oh, there we go, library welcome kit. Let me open this so I can share publicly, copy, and please, someone let me know if you're able to open it. I'm going to put it in the chat. If you're all able to open it, this contains all of the information you might need, basically all of the pamphlets, all of the different things. Somebody wanted to share some feedback.
It opened without any problem. The
Wonderful.
welcome kit is open.
Fantastic.
Okay, and I will copy that so that when we send out the video, we'll also send this information to everyone. Okay.
Amazing. Amazing. Thank you so
Yeah.
much. And thanks everyone as well.
Let me just bookmark it. Would be even better. That way I
Absolutely.
won't lose it. Okay.
Yeah, I just wanted to say thank you to all of those who spent some time with us and also those who asked questions. Again, as Lenny said, it's just a little bit of practice needed, but it's such a rich resource. I highly encourage everybody to start using it because it's very precious to have it in many, many situations. I also was a teacher for a few years, and had I had this, I say this all the time, but had I had this, it would have made my life so much easier.
Yeah. What I did note is that several people want to talk to administrators about this, to let them know the wealth of material that's now available through the JoVE videos. So that is one reason we have to get that get the information out about where they can find the brochures and all that, which you just did. But I'll make sure that goes to all 320
Amazing.
people who registered. Okay, we have any other questions? I know it gets tiring to teach for an hour plus, and you don't always see the people, and you have me interrupting you with the chat. But we were very thankful, pleased that we finally got this done, because I say this is one of those underutilized, hidden gems that needs to more people that have access to Research for Life need to know about. So I think we were successful in that sense.
Thank you, Lenny. I
Okay.
really appreciate that. And I can see somebody asking about the non-STEM materials. Absolutely. We have a new business section. We've started adding some psychology content, marketing, a lot of different areas that are in the humanities we're developing because we know it's just as important. So yeah, thank you, Lenny, and thanks everyone. It's been a pleasure. Again, as a brand director, I think it's important for me on a personal note to know that this resource gets out there, but it's even more rewarding to know that it comes to fruition in settings where people truly need them. So thank you. I hope the Country Connect has found this useful. If anybody wants any more information, feel free to contact us. Okay.
Okay, appreciate it. And have a peaceful afternoon.
Thank you. Thanks,
I
everyone.
can go look at my email now.
Have a great day. Bye.
Yeah. Bye.
Thank you and bye.
Thank you.
Bye.
Bye.
Thank you and bye.
Bye.
Bye-bye. Thank you very much.
Thank you. Bye bye.